Welcome to Hidden Layers, where we explore the people and technology shaping artificial intelligence. I'm your host, Ron Green. Today, we're going to talk about a quiet revolution happening in biology, one that I am personally extremely excited about, namely the use of AI to design new proteins to fight disease. In AI, we're used to benchmarks. We measure models against known answers, compare the scores, and often treat the benchmarks as the test. That can be useful, but biology has a way of humbling clean benchmarks. A designed protein can look promising on the computer screen and still fail when it's synthesized, expressed, folded, placed inside a cell, and asked to do something useful. That's what makes the Bits to Binder challenge so interesting. This challenge, organized by the University of Texas Austin BioML Society, asked teams to use modern AI tools to design small proteins that could help immune cells recognize and attack tumor cells. In plain English, the goal was to see whether AI could design a new biological binding component for a CAR T cell, one that could find the right cancer target and help trigger an immune response. The designs had to do more than look good on a computer. They had to express, bind to the target, and activate the associated CAR T cell in a real biological test. 28 teams spread across 40 different countries used open source AI methods to design and submit a combined 12,000 protein binders. Then those designs were tested in vitro, moving from evaluation to biological reality. That alone would make the challenge notable, but the open science approach they took is just as important. All of the software is open source, and all the designs and methods are being made publicly available with no intellectual property claims, so the results can help move the scientific field forward. Joining me today to talk about the challenge are the organizers, Kosonocky, Daryl Barth, and Aaron Feller, all of whom are PhD student candidates at the University of Texas at Austin. Together, we'll explore what happens when AI-designed biology moves from prediction to real-world experiments. I'm unbelievably excited to have you all here. I've been waiting for this episode for almost 2 years. This is going to be a blast. All right, so let's start at the beginning. I teed up why this challenge was so important in the intro, but I didn't really talk about why are proteins important and why is protein folding prediction so important? What did AlphaFold do that changed the game? Let's just start at the beginning there.
Right. So I think one of the most important things to address is that proteins are kind of the basis of biology in terms of interacting with the material world. There are micro machines that make the materials in our body in some cases or perform all of the functions that need to happen. So it's really important. And oftentimes the structure of something will dictate its function. So it's really important to be able to look at a protein and say like, oh, it does X. And we can figure that out because of the way that it looks.
Because if we know the shape, we can predict more accurately how it will behave.
Exactly.
Okay.
Yeah.
Okay.
And so that's been kind of the dark matter of understanding proteins for a while. Is like we could figure out what the amino acid sequence was, what it was composed of.
Because there's a sequence of amino acids, and then they get folded, converted into a protein structure.
Yes.
Okay.
Yeah, it happens reliably over and over, right? So you have the same sequence, it folds into the same structure every time. And actually, before the recent Nobel Prize for protein folding and protein design, you had, I think in 1923, Anfinson's dilemma. So he proposed this idea of, we think these amino acid strings fold the same way every single time.
I didn't know it went back to the 20s. Okay.
And so by proposing,
I think it was the 70s.
he won the 70s. Sorry.
Okay.
He won the Nobel Prize for pretty much proposing the question and finding some of the first information that they fold the same way.
Right.
So it's a 100-year problem.
When he proposed that, was that controversial? Do you know?
I'm not sure if it was controversial, but his idea was that all you needed is the amino acid sequence.
Because so I could see somebody saying like, yeah, but you would need the context, and the context might, the environment might play as big a role as the sequence did. But it turns out, for the most part, it's
probably somewhat true. Because there is some crazy research out there where they're testing out how proteins work in like different solvents, but that's like really complicated. And then you also have disordered proteins, which don't take on one canonical structure, and they like change conformation based on what's around it. And that's just entirely really complicated.
That's like the cutting edge right now.
I've never even heard of disordered proteins.
Yeah.
Okay. So what is AlphaFold? And like why did that affect the game?
Yeah. Well, so I feel like, we had Anfinson's dogma, and it was like, is an amino acid sequence all that you need? And then that really launched the field. And then I think in the 90s, that's when this competition called the Critical Assessment for Structure Prediction or Protein Structure Prediction, CASP, came about. And that was like the Olympics for protein structure prediction. And every 2 years, they would have structures that weren't released to the public yet. So somebody did X-ray crystallography or cryo-EM or NMR, and they solved the structure of a protein.
And that may have taken years to figure out that one protein structure.
Yeah, exactly. Yeah. And so they kept those from being released into the public, and they had everyone use the cutting-edge models that they had created at the time to try and like figure out what that protein structure was of the ones that we already knew were solved. And so that happened every 2 years since the 90s. And there was like moderate progress that was made over time, but it wasn't really until I think like 2018 or 2019 when DeepMind released AlphaFold that it just like fundamentally changed the game and blew all of the previous models out of the water.
Right.
Because if you think about it, it's a physics, if you're trying to like model exactly where all of the atoms in a protein are, it's like I still think we can't computationally fathom what that would look like in a protein. So you needed a different approach, like a transformer, to be able to like look at it from a different perspective of, can I combine my knowledge of already existing structures and the amino acid sequences that go with them to really get there. Right. And it wouldn't have been possible without the like decades of work that people had done getting these protein structures already and like figuring out.
I think that's a really important point, because it wasn't like DeepMind went and created this in a vacuum. They used all of the hard-won protein structures that had been built up over decades as training data for this.
Also protein sequences too. So there's this so we have the protein data bank, which is it's like several 100,000 protein structures, like all crystal, NMR, and so on structures. And then protein sequences have been, since the like advents in DNA sequencing technology, we've been able to figure out all these different possible and like probable protein sequences out there. And people have made models to predict what these do just on the sequences themselves. And then so from all of the sequences out there, there's like slight variations in these. And then because if two things like co-vary and like change always together, we can be somewhat we can suspect that these might be kind of like close in space because they're interacting with each other, and thus one mutation is infecting the probability of the other one there. And so AlphaFold really it's combining the structures and learning on the structures with also learning on the sequences and the coevolution of how these different amino acids are evolving with each other.
Okay.
The co-evolving residues can sort of like staple those close in 3D space because they're interacting, right? They're co-changing, right? You change a positive charge on one, you now need to compensate in the other.
That makes complete sense. Okay. So AlphaFold really showed that this was possible. The bits to binders challenge that you guys organize is shockingly ambitious, in my opinion. I mean, it's like we go from, hey, is it possible to predict amino acid sequence to protein folding to, yeah, there are some tools that can do that, to let's go see if we can synthesize, hold a competition, pure open source software, and go build a protein to accomplish a very, very specific task. That to me, it felt like a giant leap, moving from saying, hey, can we predict protein structures to, or protein folding, to creating new ones that work and do and accomplish biological tasks. Okay, so my question is like, is that where the field is now? Is it very common at this point? We went from 2018 to this, and are you just seeing an explosion there?
I think so. I think structure prediction still is not solved. I think we've gone very far, and it works very, very well for many, many cases. I think there's still room to go. So I think CASP is like, we're not shutting down CASP. Like we actually, like ImageNet and the whole ImageNet competition, like they don't do that anymore because it's like a solved problem basically.
Right.
But like we still haven't solved the protein folding problem.
Okay. So CASP is still being held.
Still very much going.
All right.
Yeah.
Okay.
Yeah.
And it's a lot of like transformer based models. So AlphaFold sort of launched its own.
I would bet so.
Yeah. Folks are now like closing the loop and re-injecting physics into these models, which was the old style. The transformer took over and won, but now to like edge out transformer only, you need to bring physics back into the equation.
We're seeing a lot of that in other fields as well, where it's sort of physics-based or physics-constrained based modeling, right? You know, if you're trying to predict how a physical object would move. You constrain it. Well, it can't go faster than the speed of light. I mean, there are simple things like that as an example.
It's like gravity exists.
Like gravity exists. Exactly. Okay.
But to jump back in, to like this sort of leap or attempting to design, there exists a lot of open source software for this, right? It's a hard problem. If you look at academic institutions, folks like us tend to like hard problems and want to crack them, right? That's why CASP has been so successful at pushing the edge. So you see a lot of open source models that are coming up built on sort of the predecessor. So how do you combine these different workflows? How do you screen, identify, use the structure, prediction as part of the modeling? So a lot of tools existed. A lot of manuscripts have been published on the topic of de novo design. So the field is growing. We sort of have caught this wave, right? You see folding sort of be solved, as people say, and then, this growth of a lot of attention, a lot of attraction, the Nobel Prize, and a lot of labs trying to work on this. So it felt right to run a competition to see how well these systems worked.
Yeah. And I think, so in 2024 is when we really formulated this whole thing. And at that time, we had several ongoing projects with people in the lab and other labs where we were trying to design proteins to solve a given task using the models that were open source and state of the art at the time.
And I think one thing here is that diffusion models were really becoming a big thing at that time. And so that enabled protein design instead of just protein structure prediction. And so that was kind of,
Totally.
Yeah.
And what's actually funny is that a lot of the new protein structure prediction models are also diffusion models now.
True.
So it kind of like has, it's influenced everything.
Okay.
But yeah, so like around that time in 2024, we like didn't really know what worked best, and we like wanted to find out like for our own sake, in some sense, for the projects. And I think a lot of people in the field are wondering the same thing. And we kind of just like, so we have the BioML Society, which do you want to go into like the origins of all that?
I've got the organizers right here.
Oh.
Let's see, how do we start, Aaron?
I mean, it was a group of students who were interested in this space. This was before ChatGPT came out, right? So the world at large didn't have a focus on AI. So we were interested, wanted to apply it, had seen AlphaFold 2, I think is what drew my interest. And we were sort of isolated. There wasn't a lot of knowledge around. And so we had to focus on these specific labs working on these problems and read the papers and try and like wrap our head around what they were attempting and how these models worked. So we really started it as a group to teach each other and improve it at modeling in this way.
Totally. And so we started this in January of 2023. And then we initially just had some guest speakers come by and like all just like talk and try to learn with each other. And then in the fall of 2023, we started a lecture series actually on applying machine learning to biological problems. And we were all lecturers in the series, and as well as some others too. And that was super fun. And we were kind of just like learning as we went. And we attracted some people to the organization and like had a really fun base that started to form. And then in the spring of 2024, we were kind of just thinking about ways to engage the community again in some way. And then I really don't remember who came up with the idea at first, but someone was like, we should do a hackathon. And then we're all like, sure. And like didn't really know what that meant. I don't know.
When I heard about the hackathon, I was blown away because I didn't know that there were open source tools that could even do these types of things. I knew that, I knew about AlphaFold, but I didn't know about the other tool sets out there. We've talked about this before, but sort of the skepticism within biology, maybe around the use of machine learning tools or AI tools. I want to talk about that a little bit, meaning, I remember talking to one biology student a couple years ago, and I was saying, what's the reception of computational biology within the biology field? And he said something like, well, you get on board because the train is leaving the station. Like that is the future, but it's not always been necessarily well received by professors with, lots of years of experience. They may have some skepticism. What's it like right now? It's 2026. I remember when AlphaFold came out, some people didn't believe that it was accurate or it could be improved. Is there a general embracement of computational biology, or is there still a lot of skepticism and resistance within the biological domain?
I feel like it can be on either side. A lot of or like the bubble that we live in, people are pretty receptive to it. The biggest thing is they want to see whether it actually works in reality. So you need to like print it into carbon is a statement that one of our PIs says a lot. And so there's a lot of skepticism if someone just publishes a model and there's no experimental data to back it up. That seems to be one of the biggest ones. But I think people are really impressed at the like speed at which you can come up with potential hypotheses. So that seems to be a thing that pretty much everybody is on board with now, and people are trying to like incorporate more AI into their work for sure, with this very skeptical hat on of like, we want to see behind the scenes and make sure all of the code is working as we want to do it and that type of thing.
Right. Well, all right. So that's a perfect segue because, in my world, when I'm building AI models, for the most part, we don't really have to worry about the difference between, the digital domain where they're being trained and where the data is being fed in, and then some transition to reality, right? The real world. We can essentially treat the benchmarks as the final test, right? Because they work really well. That's not the case with biology, right? It is entirely possible that you could develop an amino acid sequence. It looks like it's going to fold and bind and do really well, but the reality has to actually be tested. So a couple of questions there. How big is that gap? Like, what did you learn as a part of this challenge? Is the gap bigger than you expected? And is it, and to close that gap, is it really a question of just getting better at being able to simulate biological systems in the digital domain, and then you could see a path, maybe, I don't know if it's a year or 10 or 100, where you do all of your research and development purely digital?
That's a very big question.
Yeah. Let's see if I could take a shot at that. I mean, so going back to this concept of like, we have to go print it into carbon.
Yeah.
We can't use the benchmarks, right? So in modeling for proteins, you have certain evaluations you can do in silico. And this is like model perplexity during generation of that amino acid sequence that would fold, or how confident the model is. So AlphaFold has like a confidence metric of how confident it is of that structure. So you can look at all these data points and try and say, yes, this is a good sequence. It'll fold this way and it'll perform this function. But it's sort of like running Claude code, except you can never run the code. Right? You're just saying, okay, it generated these tokens in this order. Here was the perplexity during that generation. We'll pick this script and we'll ship it. Right? You need to run the code to evaluate whether it's working or not. So that's sort of the wet lab side of things, is running the code, if you will. So we have DNA code, right, that gets converted into proteins and then they get tested in this real world scenario. So going full computational, I think things will get better. We'll have better hit rates, better success rates, right? The field, it ranges based on model and task from, less than, let's say, 1 in 1,000 up to, some papers report like a 70% success rate on, let's say, improving an antibody's binding.
Okay.
Right? But it depends per target, per antibody you start with. There's so many factors
There's
involved.
Just so many variables.
Yeah. So that's, that's what we were trying to assess.
Yeah. There's
the competition.
There's one target for binding in particular called TNF alpha that like most models cannot make binders to. I think the only one group, Chai2, has been able to actually make binders to this, as far as I've seen. And it's just like a weird target that forms like a trimer, it's called, with each other. So it like has this very strange interface. And for some reason, that just lends it to be very tricky to make protein binders against.
And is that because that type of structure is uncommon and so the models just haven't seen enough of it? Or is there something deeper going on? Or is it hard to know?
Yeah,
I think it's hard.
I think it's hard to know. Yeah, I think it's hard to know. Because I think there's so many objectives in biology, and we don't have like amazing suites of data for each of the objectives. So it's hard to really tune that. But to kind of get back to your question, there's a lot of funding going into building virtual cells now, and like creating these types of digital environments where you can model. So that's like definitely the direction that we're going in. But I don't know how long it's going to take.
Yeah. It's funny. I've lost track of the times I've talked to people who are in sort of AI biology startups and the goal. And I'm like, what's your goal? And they're like, like, well, we're trying to figure that out. It's like, just start and then we'll figure it out. One of the things that we're seeing more and more at the bleeding edge of AI is there's something called sort of the verification dilemma. It's like coding agents, AI coding agents that can actually write, programming code are far ahead of pretty much every other domain because it's verifiable.
Yeah.
Mm-hmm.
You can generate some code and then generate a test to see if it works, or have tests beforehand. And you just don't have that in biology right now. You have to go through all of the complexity of the wet lab, right? And as you said, actually creating the carbon. The challenge was over 5 weeks. Is that right? And then it took almost a year to synthesize everything. So, I mean, that right there just speaks volumes.
Yeah. So I think challenge was in August of 2024. We collected all the sequences in October, I think. So like 5 or 6 weeks after. And then we, filtered them, sent them off to the DNA synthesis provider. I think we got the sequences delivered to UT in January. And then we sent them off to the partner company that we worked with, Leah Labs. And then they were fantastic, and they worked so hard on this. It was amazing.
Hard.
Yeah.
Thank you, Leah.
And just to say this validation, we tested 12,000 sequences in pooled screens. So because of the assay that they had set up and what we had available, we could test 12,000, right?
And I think the cells, so we would test 12,000 in a pretty high throughput assay, but it still takes like weeks for these cells to grow.
Right.
And like you have to do it in like multiple replicates in case there's weird things happening in the biology. So you have to like make it consistent. And then like we did the pooled high throughput screening as the first main filter to see how things worked. And I think in the end we had, I want to say, a 6 or 7% success rate roughly across the board. Some teams had kind of like lower success rates, and then some teams had, I think, up to like 38% success rate from the high throughput screen, which was very, very high. And then so we took after that like the top 10 designs total in terms of performance in this assay. We tested them across a broad range of different like immunotherapeutic functions. Which should we actually just go more into?
I would love that. Yeah, let's jump in.
Cool. I guess I'll lead that. So basically, the format for the competition was in this immunotherapy called CAR-T therapies, and this stands for chimeric antigen receptor T cell therapy. And so our bodies have several types of immune cells, one of which are called T cells, and these are sort of like the policemen of the body. Like they're like going around to these different cells and trying to figure out like if these other cells are infected or not, and if they are infected, then they're gonna or like not doing well in some way, they're gonna try to eliminate them and try to like make the body overall very healthy again. And we can take a few decades ago, someone proposed that we can essentially engineer these to recognize not infected cells through the normal mechanism, but instead to recognize any arbitrary like cell or protein. And they do this traditionally by attaching antibodies to the cells to kind of guide them and have them stick onto different parts of different protein surfaces. Mm-hmm. And so this is like a huge therapeutic field, and it works actually really well to cure certain types of blood cancers. And so it's like very exciting and promising, and people are very much like wanting to explore all this stuff further. And so Leah Labs, their company, I think they're mainly applying this to cure blood cancers in dogs. And so it's like super just like awesome stuff. And they developed this high throughput assay to screen like thousands of these different like CAR T cells at the same time by figuring out when they detect the target that they expect, is it going to cause them to grow and proliferate more than a control, than something not being there. And so the reason why we expect this is because the body has this mechanism for these cells where if it detects the target, then it's going to make more copies of that cell to then better mount a response, because you have more opportunities to actually have everything work correctly. And so we can measure all that with DNA sequencing technologies and basically get some like pretty clear like plot that is like, if it worked, it's on this side. If it didn't detect it, then it's on the other side.
Okay.
Yeah.
Okay. And the goal would be ultimately to essentially elicit an immune response that kills those cancerous cells.
That's
conceptually.
The goal, yeah. Mm-hmm. So the cancer cells contain a specific protein called CD20, these specific cancer cells we were looking at. And the goal of the competition was for people to design proteins that can bind to this other protein, that when we integrate them into the CAR T cell, it'll guide the CAR T cell to that specific cancer target.
So when you're thinking about cells in the body, a lot of them have like kind of protein to peptides. So those are like small sequences of proteins that stick out on the cell surface. And so a lot of like existing cells, they have the MHCs. So they're kind of this, we don't need to
major histocompatibility complex.
Major histocompatibility complex. We can probably cut this.
No.
This is way too intense.
This is way too intense.
No, we're totally leaving that in.
Okay.
No, we're going to leave that in. No, keep going. This is awesome.
But it's essentially like a homing device. And so when you have sick cells, you can, in the MHC, they're going to put out peptides that are going to be like, I am sick.
Okay.
And so
it's like, here's my sick maybe peptide.
Okay.
But if you have like a blood cancer or something like that, you don't have the I am sick signal.
They shut it down as part of their progression to forming a
that
cancer.
makes total sense. So you're essentially trying to create that signal, that missing peptide indicator.
And
so rather than targeting the peptides the MHCs hold on the surface, we target CD20, which is
another
present
peptide
on this cancer.
Yeah.
Okay.
Or protein.
Another protein that's
okay. That makes total sense.
Mm-hmm.
Okay, so I know everybody had to submit amino acid sequences, and they had to be 80 amino acids in length exactly, not more, not less. Why?
Yeah, I think that was mostly just chosen to essentially just make it really scalable.
Okay.
Because we would just have like the set sequence length. Everything else could be synthesized by Twist in terms of like just the blocks, and then they could like assemble them together.
Okay.
And it would be consistently sized, and everything would work out well that way.
Okay.
But I think 80 is, at least we thought it would be enough to create binders that could target it. So it's
long enough to probably have the complex functional behavior you wanted, but not so long that it got too difficult to do the wet lab processing or whatever downstream.
Right.
Okay.
Right.
And then, sorry, I'm kind of curious. What was actually the hardest part? Was it what you thought it was going to be? I look at this and like, was it making something that folds? Was it making something that binds? Was it making it express in the cellular context, or was it that last step of making it trigger the right T cell response?
Yeah.
Right,
I think I'll talk about this one. So after we did this whole competition, we spent like months analyzing the results. And we were at first looking at just whether in this high throughput screen, whether the cells divided more, proliferated more or not. And we were checking, that's basically like our true positive signal, like these probably worked. And then we spent a lot of time looking at that. And then we realized that actually like half of the designs didn't even like result in really like functional T cells at all. And so they kind of just didn't produce like a proper protein. And so they just didn't even express correctly. And so basically my answer to your question is that all of the above are still very difficult. And we're still trying as a field to like disentangle all the rules that make these proteins either express better or, target correctly, and try to just like bump up the success rates at each of these different steps, and also be soluble, and also not have, like most people haven't even explored this whole topic of these proteins not having immune responses and stuff. And so like,
this is a great example of the multi-objective.
Yeah.
Like there's so many
Yeah.
failure modes, and it's hard to disentangle. And so then because it's hard to disentangle from the assays that we're getting, you don't have the validation data to throw back into the model to like then train on those objectives. We have like, to some extent, we know which ones didn't like proliferate. And like, so you could, I don't know, I guess there's a couple reasons why they wouldn't proliferate as well too. So yeah.
Right, they didn't fold, they didn't express correctly, they weren't presented on the surface of the cell. All these processes have to succeed for even the protein to sit on the surface. Okay, now you have a protein there. Now does it bind its target? Okay, once it binds, does it activate a response in the T cell that causes the proliferation? So that interaction like sends a signal to say, hey, start proliferating.
And what's even crazier about that is that if it binds too tightly in T cell biology, it can actually cause the T cell to like kill itself, because it's like we don't want to have super hyper overactive T cells, and so that's like a natural like dampening mechanism in in so they had to like they bind in the Goldilocks zone for it to actually work, which is like
just
so
a simple
crazy.
1,000 things have to go.
Exactly.
I mean, it's sort of this thing where, okay, let's say like, I mean, the success rates were maybe, you said 7%, I think, just overall for proliferation, right? So
no. So, sorry, that's totally correct. Yeah.
So let's say it's 10%, and you've got 4 hoops to jump through, and they all work at 10%. You go 10%, 1%, point. Now you're getting into small numbers.
Exactly.
Yeah. And so we, a big part of this was afterwards, we wanted to find out like, what caused some things to work better than others, and can we disentangle some of these things? And I think the main findings that we found, which we discuss more in the paper, is that there's these generated proteins look very different from like regular proteins in terms of the sequences. And our hypothesis that we come to is that these sequences contain like different amino acids that are causing, weirdnesses with the rest of the biological system that maybe is influencing them not expressing properly.
Okay. Oh, and so you really wouldn't find these naturally for that reason.
Some of them.
Some of them.
Yeah. Some of them.
Okay.
I think the story is probably more complicated.
Yeah. Exactly.
Yeah.
Okay.
And
yeah.
Okay. I wanted to ask you about, this was a student-led competition. I found it to be just astonishingly ambitious. I already mentioned that I didn't know that there were, so many open source packages capable of achieving these protein fold prediction results. And I know each team got to, choose their own. Do you think that you guys were able to move so quickly and be so ambitious because it was a student-led organization? Do you think like if you were in a lab or if you were in a company setting, there would have been more constraints? Would you have to have moved slower? Or is it the exact opposite, that you were hamstrung because of, maybe you just didn't have the same amount of money and resources to spend on this?
Oh, that's interesting.
I think,
I
was going to say, I don't think we realized what we were getting into when we started this.
100%.
That's how great stuff is accomplished.
You don't know how big you're shooting until you're too deep. You can't turn back. And you're like, well, we got to finish.
Yeah.
I mean, we were really fortunate. So much partnership with different companies.
Yeah.
DNA synthesis, I think, was half covered. We had Lea Labs working with us with their assay, putting hours in. Small little startup company. I can't think of the other, like we had reagents donated, like so many different pieces. So like money, I'd say money was an issue, right? We were an academic lab. We didn't like have our own funding for this or win it. So we kind of had support of the community. Mentioned earlier, like the professors around us really supporting the whole ML push. I say ML, AI, at UT here. So we're fortunate in that way, and they were on board with that. So like special place, special time. We had the club. And then being in an academic space, I feel beholden to do things open source, right? I'm funded by taxpayer dollars.
Right.
I feel like everything I work on should be released for public use. And there's some exceptions where you need patenting for things to move into production. But as far as exploration of ML and generating
that's one of my
data,
favorite parts about this challenge was it was completely open science. I mean, it's really kind of striking. Everything was open source from a software perspective.
Yeah.
To compete, you had to make no intellectual property claims on any of the sequences or the methodologies, right? I mean, it was just all about advancing the scientific field.
It was all very intentional throughout the whole design process.
And I think that enabled us to get a huge diversity of ideas and models that you probably wouldn't be able to get if you were in a company. Like you could pull open source models off the shelf, but the order in which you put them together, or, like the other bespoke platforms that people had put, all across the world.
Yeah.
Yeah, you wouldn't get that. And we were just looking, we had a team that was like someone from Boston, someone from Uganda, someone
so
UK.
It was all over the place.
So it's great. You
the
get this crazy diversity of ideas
and South Korea and
collaboration.
Spain and India.
South Korea, yeah.
And Germany.
one team.
On one team, yeah.
Okay, so I have to ask, who were the winners of the competition?
Yeah. So we had two main categories for who won. The first one was in the high throughput screen, and this was more like who had the best hit rate overall in this whole thing. And so from the high throughput screen, the best hit rate was Nucleate UK London, and they had a 38% hit rate of their submitted sequences, which was like pretty much astoundingly high, honestly. We were pretty shocked by that. And then after that, so from that screen, as I was kind of mentioning earlier, I think we took the top designs from some of the teams. We took 10 different designs and then tested them individually in a series of different like T-cell functional assays. And so this includes things like cytotoxicity, like whether it can specifically kill the cancer cells compared to like a muscle cell control. It included things like whether they release the proper cytokines in the right amounts and whether they can expand and proliferate and like do these things that we expect healthy functional CAR T cells to do. And so from this, we took like a weighted average of these different individual functional assays that, of course, Leah Labs were the ones who did all this amazing work. And from that, the top team was the Perez Lab Gators, and then they're from the University of Florida. And then close behind them was this team called Amigo Acids, which is a fantastic name. And they're the team that had all the different competitors from around the world, which was super cool to see this team kind of like spread all these different countries around. It was super cool. And then the third team was the Schoder Lab, and they're from Germany, and they were also like had a very strong design. So it was some
awesome.
good successes
What did the
overall.
winning teams win?
Oh, yeah.
Some trophies.
Yeah, they got some trophies.
Some 3D printed protein trophies.
Yeah, so we worked with this other this person from, I think, the Maker Lab is what their name was, and they created these awesome trophies. We made trophies of CD20, the protein, with each of their binders like binding to it, which was
the classic
so
like
cool.
Ribbon image of the backbone of the protein.
Yeah,
it looked really good.
And then we sent them to them like a month or two ago.
Yeah. Oh, that's awesome. Okay, final question. What are your takeaways from the competition? Did you leave it on a personal basis more excited about the future of AI intersecting with biology, or was it a disappointment? I just any final thoughts on the experience?
Do you want to start?
Sure. Yeah. I left pretty excited about it. I mean, the fact that we got anything to bind and to grow and like go through this really hard task, I thought was really exciting. And I think for me personally, it was just so much fun to be a part of this international competition and see the excitement and the buy-in. And I know, like, the internet connected everybody. And so this was kind of a biological cognate of that in a way, because I feel like a lot of times when you're in wet lab, you're interacting with someone 5 feet away.
Yeah.
And so this sort of enabled like collaboration across borders. So I think, for me personally, I just loved seeing people tune in from everywhere and having them form teams that are so multidisciplinary and seeing all of the variation of ideas that came in. So
that part was really, really cool. And I'm really looking forward to meeting all the people that entered the competition at like conferences or just like around. And I've met a few of them in the past who have like happened to be in Austin at the time. And so we just like caught up and got coffee with someone who entered the competition, which was super fun. I think I agree with Daryl. I'm very excited about the field from this competition. And I honestly think we've like uncovered just a lot of different questions on like how to make this better and like questions that we need to address to really like bring this as to become a more mature kind of field overall. Like what are these weird protein sequences that don't even look like biological sequences doing? Like, are we like, how is that impacting biology? What are the mechanisms behind what's going on? Like, what are the mechanisms behind these things working or not working? And like we only really talked about and really measured protein binding. And I'm really excited for the whole future of like enzyme design, which it's really starting up, making proteins that can catalyze reactions. And I think it's extremely bold, but people are even trying to make like new to nature reactions that they're trying to design and to like catalyze like brand new materials into existence, which is super, super cool. And then like even further than that, there's this whole arm of like making proteins that have dynamics and that like change and making like full-fledged protein machines. And I think there's a lot of work to be done before we can get to that point, but I think it's super cool. Yeah.
Wow. Any final thoughts?
Yeah, absolutely. I totally agree with both of y'all's points. Seeing people come together, seeing like very young PhD students, maybe in their first year, who are interested in this space, join a team, submit some designs, be able to work through the workflow to provide a space where they have like an end goal and there's wet lab validation, I think was like, for me, one of the coolest things of running the competition. I'll throw a little pessimism in.
All right.
I'll be the one who throws some pessimism, but our hit rate was quite low. None of our designs outperformed the positive control. I say our designs, but the submitted designs, right? We chose CD20 because there are clinically approved CAR T therapies for this, right? So we had a positive control to benchmark against, right? Okay. We didn't reach that, but imagine one team 5 years down the road, and they've they can do 12,000 from their design that's improved, from the data that we collect in this competition or similar things, or just like the large global growing scale. So I see a future. I see definitely like this contributing to that, but like right now, everything's a little bit scattered. There's so many different methods. Each lab or each researcher benchmarks their own method on their chosen targets, right? There's no CASP. There's no like critical assessment of these methods, head to head on single tasks one by one. So, we did this one competition. I could see something like this growing, other folks taking up the mantle and running something that repeats so that, the kind of open source academic community can really hone in on what is working, how to make it work better. And then, across all these checkpoints of folding, binding, proliferating, different evals, different filters. So it kind of told us where things stand today.
I love ending on a realistic note there, because I think, with much of AI right now, we can see just how good it's going to be. But we're also living in the world where, it's still prone to failure in ways very frequently that's kind of unpredictable, right? It almost has that sort of human element of unpredictable failure points, right? But I really do believe that the competitions like that you put on are so incredibly important for science, because you had so many countries and so many teams involved, and that experience is going to give everybody involved the optimism that they can do these types of things going forward. And just being willing to step up to the plate is half the challenge in life. So unbelievable job, major kudos. You should be really, really proud. And I can't thank you all enough for being on the podcast today. This is awesome. I've been looking forward to this for a long time.
Awesome.
Also, thank you for helping us like ideate early in the whole competition. I don't know if you've mentioned that you were talking to us early on about this.
Hanging out for office hours.
I couldn't have been happier to be involved. This is just an absolute joy. Thank you all.
Yeah, thank you.
Thank you for listening to Hidden Layers. This series is hosted by Kung Fu AI, a management consulting and engineering firm focused exclusively on artificial intelligence. If you have any questions or thoughts about today's episode, or if you know someone we should feature, please visit us at kungfu.ai.